Hello and welcome to this showcase session, which is designed to give an opportunity to interact with experts in nanopore technology for conservation. Each of our speakers will provide a brief overview of their research and interests. They will then join me in a general discussion. We'll also be inviting questions and opinions, and as this is an on-site only session, we'll be taking questions from the floor. I'm now delighted to welcome our first speaker, my colleague, Cara Dix. Cara is the Program Manager for Conservation Genomics at Oxford Nanopore Technologies. Please welcome Cara.
Thanks, Dan. So we are in a biodiversity crisis, and over 44,000 species are threatened with extinction. ORG.one is Oxford Nanopore's, it's an initiative by Oxford Nanopore to support conservation and expand genomic resources for threatened species. We aim to do this by supporting local sequencing where possible, so these species are sequenced in their countries, and by promoting open access data and developing an active and distributed community of conservationists, researchers, and bioinformaticians working together and sharing their knowledge. Already we've supported over 90 projects, and ORG.one participants receive flow cells and kits to sequence the world's most threatened species. These are species categorized by the IUCN Red List as extinct in the wild, critically endangered, and just this month we've expanded to also include endangered species. We've also partnered with New England Biolabs to provide even more sequencing reagents so that the cost to participants is reduced even further. We're really excited today to be celebrating 10 years of the MinION and 10 years of London Calling by sequencing the genomes of 10 species, from the Asiatic lion to the Cuban crocodile to the extinct in the wild Socorro dove. And we're really grateful for the support of the European Association of Zoos and Aquariums Biobanks for providing the samples, and to our partners here on stage today. Thank you.
Thank you, Cara. I'd now like to welcome our second speaker for this session, Heather Ritchie-Parker. Heather is a research scientist at the Royal Zoological Society of Scotland, where she works within the WildGenes team. Her work focuses on providing bioinformatic analysis of genetic data to government agencies, conservation charities, and zoos across the world. Heather works across a variety of species of conservation concern, including the northern rockhopper penguin, the Rote Island snake-necked turtle, and the pine hoverfly.
RZSS is a conservation charity, and the RZSS Biobank is one of 4 of the EASA biobanks across Europe, and it's a facility for zoos and aquaria across Europe to biobank samples to be used as genetic material. So one of these biobanks is in Edinburgh, and it acts as a repository for samples that have come from institutions within the UK. Having these biobanks is a really important facility, both for individuals that are in our ex situ collections, so our captive collections, but as well as their in situ wild counterparts. Often, genetic material is really difficult to come by for species of conservation concern, and the amount of genomics resources available for these species is often depauperate. So this is where our zoos and aquariums can act as a really important place to get high-quality genetic material that can then be used to produce these resources that we don't currently have. And what's been really exciting is being able to produce some of these reference genomes through the ORG.one, and that is really going to be a fantastic resource both for myself as well as other conservation colleagues. And these are going to be a range of different species of conservation interest, from the Cuban crocodile to the Amur leopard and to the northern rockhopper penguin. Now, the northern rockhopper penguin has a special place in my heart, and not just because they're endlessly charming, but because they're also an endangered species. And RZSS and project partners are working to conserve these birds in the wild. And having a high-quality reference genome published through ORG.one is going to allow us to have some really nice high-quality data analysis, and that's going to help inform conservation practices in the wild. Thank you.
Thank you, Heather. I'm now delighted to welcome our final speaker, Ben Price. Ben is the Principal Curator at the Natural History Museum in London, where he's responsible for part of the collection of 80 million specimens. His research focuses on computer vision and high-throughput genomics for biodiversity discovery. He leads part of the Horizon Europe-funded Biodiversity Genomics Europe project, sequencing modern and century-old specimens to fill gaps in DNA reference libraries and enable DNA-based identification for all. He's also the owner of the longest sentence. Ben.
Thanks, Dan. Yeah, sorry about that. Brilliant. So thank you very much, Dan. We're here talking about a couple of projects where we're using nanopore sequencing in gene sequencing and genomic sequencing. So it might surprise you to learn that we have names for about 10% of the species on this planet. And for most of those, we just have their name and we know almost nothing else about their biology. Imagine trying to do chemistry with 10% of the elements known to you. So I've got a lovely plot up there of a periodic table for biodiversity. And you can see that each one of those squares represents 100,000 species. So all the vertebrates, all the things we typically care about, would fit into one of those squares. And it just highlights that there's a huge amount of unknown biodiversity out there that we need to accelerate our exploration of. So one way of doing this in a high-throughput way is through amplicon sequencing, DNA barcoding. And the International Barcode of Life Group initiative, at least, is a global initiative to help register all species using DNA. Within the UK, a sub-project of that is called Bioscan UK, and they're planning on sequencing, or in the process of sequencing, 1 million arthropods from across the UK to look at the biodiversity of the UK and also the interaction of these different species. And now, the reason we can do this is because with nanopore sequencing, anyone can set up cheap, high-throughput sequencing labs. You can sequence a couple hundred specimens on a flongle up to thousands and thousands of specimens on a MinION. And I'm happy to announce that today, right over there on that gridiron, we have 50,000 individually indexed specimens from the Bioscan project being sequenced live right now. Shifting gears a little bit to genomics, last year at the London Calling 23, we were happy to sequence a large copper butterfly, a UK species, subspecies that went extinct about 200 years ago. We're now exploring reintroduction of that a subspecies from Europe, and so we used 2 days of sequencing basically to generate a reference quality genome at London Calling 23. And now, since then, we've been using that reference genome to explore the population of this potential European species that we're going to reintroduce, and then also comparing museum specimens from before the extinction event to try and understand how different these two subspecies were and potentially how successful this reintroduction would be. And all of that is made possible by nanopore sequencing. So thank you.
Thank you, Ben. I'm going to stay sat down now. So we'll now invite questions from the floor. Does anybody have a question?
I would like to ask, what is the pipeline for the ORG.one when you decide how to sequence, to make the collaboration with the individual? And so what is the pipeline and what is the expectation? What is the timeline expectation?
Excellent question. So if you have a sample that meets the criteria, it's the right kind of species, and you're really thinking about actually how can we do some sequencing here, it's firstly a question of going to the website, org.one, type that into the search bar, and you'll come up with the website, and there's a register your interest page there. Once you've registered your interest, the next question is, are you able to sequence it yourself, or do we need to tap into that community and find a sequencing partner to help that happen? Because some individuals might have access to the samples, but not necessarily the sequencing capacity themselves, the knowledge, the experience to actually do that. So we have a number of sequencing partners in some parts of the world, and if we don't, we'll try and find somebody in that region and use the community to try and do that. Once we're sort of all ready to go, we've got 6 weeks once you receive the flow cells to do the sequencing and to upload the raw data to an open access database. But that's not an assembled genome or anything like that. That is the raw data. So we're not asking anyone to assemble a genome in 6 weeks. That would be quite a feat.
Thank you. Thank you. Any more questions from the floor? Yes. Tim?
Thank you. This is for Ben Price. I was wondering if you could share some of the challenges, but also the opportunities you have in using museum collections as your sample source?
Yeah, so I guess a good question. One of the major challenges is the short fragment lengths. So we're lucky if we get 50 base pairs out of some of these things. But the opportunity, I think, is really that museum samples are the only representatives that we have going back in time for those species. And we can really travel in time by sequencing those samples. It's just a challenge to get good quality DNA out of them, and you need to sequence a lot to try and get rid of all the fungus and the contaminants and the human that was handling them and all that sort of thing.
Thank you, Ben. Any more questions at the moment? Doug?
Yeah, how are you looking into integrating this into the worldwide databases? Are there any collaborations set up with the Darwin Tree of Life, ERGA, these kind of initiatives? Or?
So what we're really targeting with ORG.one is species that need action really quickly, need conservation action, and so that's the most threatened species. So it's really about getting all of this data super quickly and enabling the next stages of conservation and conservation genomics to happen. But all of the data is uploaded. We work with the ENA at EMBL to upload there, but as long as the data goes open access, so all of that data is available for anybody to use. And so hopefully there will be opportunities for that to be integrated into other pipelines as well, where it's useful.
Thank you. Any more questions? Yeah, Martha.
Do you target a certain particular quality of the assembly or a certain particular quality of the sequencing already?
So I think, as kind of Heather was touching on earlier, samples for these kinds of species can be a whole variety of different quality types. Some of the really, really threatened species, it's really difficult to get extremely high quality samples that you need for extremely high quality genomes. But what's necessary for being able to do conservation genomics is not necessarily your ultimate gold standard, platinum standard quality genome, but it's a draft reference genome. So we might be looking at a much lower coverage of the genome. We actually target around about 20x coverage of the genome to develop a draft reference genome. But I don't know, Heather, you've done some work with conservation genomics and working with different quality genomes. Does that fit with your experiences?
Yeah, I mean, I think it's one of those where it's always the better it can be is the better is what you want. But for a lot of these species, we just don't have any kind of reference at all. So without any kind of base reference genome, we just cannot do any of that work. So for conservation purposes, anything that's even draft level is really, really useful for conservation work.
I think it depends what questions you've got as well at the other end. You know, if you need a really high quality genome for those questions, that's what you need. But for the vast majority of conservation questions, like you say, it's something that's needed.
Any further questions? Heather, I have a question for you. Are there any particular reasons why you've chosen these species here today?
That's a good question, because just about every species will have a different answer for that. So how long have we got left?
Got 1 minute, 51 seconds.
Oh, well, we can't go into all of them then. So for all of them, they will have been identified as species that are of conservation concern. So the Rote Island snake-necked turtle that you mentioned before, things like that are, like that species is extinct in the wild. And collections in zoos and aquarias are insurance populations for species such as Rote Island that has gone extinct in the wild. So having references that we can use to look at the diversity of those individuals still within collections and their suitability to be put back into the wild is really important. And they'll also be used for the dark border beauty moth, which I think is one that is currently sequencing as we speak. That is a breeding program that's being done at the Highland Wildlife Park. So having a reference genome for that allows us to check that the breeding program is capturing all of that wild genetic diversity and is being maintained through that breeding program. So a whole host of reasons why samples are chosen.
Super, thank you. Just before we start to wrap up, is there anyone you need to thank today? Do you need to thank anyone?
I need to thank the Sanger Science team who did 100,000 PCRs to get that pool ready. So sequencing is now the easy, cheap part. It's preparing the samples for that. And I need to thank Jordan, who's sitting in the audience, Jordan and Dan, who prepared the 10 species that we're genome sequencing today. Thank you, Dan.
Thank you. Thank you very much. So, unfortunately, we've run out of time. Thank you very much.